So this work is done with Vijay and Vishal, who are here at ICTS. Uh, okay, so I want to start with the idea of how during development, such as in zebrafish here, um, pattern formation, change of form, and growth are all happening in tandem. And it is important to understand how these processes talk to each other. So one example when these couplings become important is when patterns themselves are growth induced, which is intuitive in the case of these wrinkling or folding patterns. And another example is during somite formation. So somites are these precursors of the skeletal fragments that come up later in this movie. The fact that they come up at the right location and are of the right size is because there is a tight coordination between growth in this region and gene expression waves coming from a clock. So this is the famous clock and wavefront model. Um, so clearly it is important to write uh, physical models that couple these phenomena and explore these couplings. So what I want to do in the talk today is to focus on the growth aspect, where the question that we are trying to answer is what are the emergent growth patterns in our tissue resulting from patterns in scalar vector tensor fields in these minimal physical models. Okay, so the kind of growth curves that we are used to are of these type, where you grow for a while and then you stop. And this is what is called determinate growth. And this is typical of birds, mammals, and in fact, most vertebrates. Um, uh, Determinate growth is also characterized by very tight regulation, by which I mean, for example, in the fruit fly, if you do a perturbation, if you do a mutation in the wing, where you make the cell numbers in the posterior region lesser, the tissue responds by increasing the individual cell size so that the final size and shape of the structure is almost maintained. So there are these compensation processes happening. And similarly, regulation also sometimes manifests as proportionate growth, where the relative sizes of tissues or organs with respect to each other or with respect to the whole are maintained. So that's what's called determinate growth, where you have these kind of growth curves and you have very tight regulation. But there are also organisms like planaria. If you keep feeding them, they keep growing, and if you starve them, they actually shrink. So the growth curves look like this, where there is really no notion of a final size. And this is what is called indeterminate growth. And there is some evidence to believe that the more ancestral species in the evolutionary tree show indeterminate growth. Okay, so these are the two phenotypes observed in biology where with these two kinds of growth curves. And the question that we are asking is, what is the mechanistic basis of these two phenotypes? And in particular, can they arise as two phases of the same mechanistic model? Okay, so let's write down the model. It's a very simple hydrodynamic model where you have this blob of material sitting on a substrate, and the material points are labeled by their position vectors, which evolve in time given some flow velocity v, giving you the new configuration at time t. The Lagrangian displacement field is defined like so, as the difference in position vectors at time t and the initial time. But what we want to do is to write down everything in the current configuration. So one can show that if this velocity is such that div v is bounded, the map from small x to cap x is invertible, and you can write down the Eulerian velocity field corresponding to this Lagrangian, Eulerian displacement field corresponding to this Lagrangian one, which evolves like so, where dt is the material derivative. So these two equations fully determine the kinematics of the problem. We need to now define the stresses in the model. So this material is composed of cells, and cells have junctions that have these proteins that lend elastic character to the continuum. Then these cells are also constantly rearranging, which gives rise to some viscous stresses. And finally, these cells divide and die, which gives rise to these active stresses. In general, these active stresses can depend on higher order orientational degrees of freedom coming from oriented divisions and so on. and also they can depend on morphogens that are implicated in growth control. So all these three kinds of stresses enter the force balance equation, which in the overdamped limit has this form where basically friction against the substrate balances the divergence of the total stress. So for now, we will neglect the Q and C dependence of the active stress and propose an active stress that only depends on the density field, and the cell division axes are isotropic in the plane of the tissue. So to close the model, we need an equation for this rho, which is again this number balance kind of equation where rho changes because of this dilution due to growth and because of birth death. Boundary conditions are prescribed normal stress at the boundary. We will change this when we want to make the system fully autonomous, and we will make it an isolated tissue. So that's the model, and the question is, what are the emergent growth behaviors that this model shows? So it turns out that, okay, so first is the passive limit of the model. There's no activity, so we neglect this active stress, and there's no density field for now. So we see that there are two special solutions in this model. In the first solution, the length of the domain grows for a while and then stops, and one can analytically compute this steady state length. So it's essentially a scaled version of the initial length with the scaling dependent on this ratio of the applied boundary stress sigma B and E, which is the elasticity of the tissue. So from this, you note that when this ratio sigma B by E is 1, this length basically diverges. So what happens when sigma B by E diverges and goes greater than 1? If you do a linear stability analysis of the system around this state, you see that this state loses its stability to the other special solution when sigma B by E is greater than 1, which is where the length keeps growing linearly in time. So it's a perennially growing phase. The growth rates can be computed again in terms of the material parameters analytically, and this is a boundary driven growth. So growth is primarily at the boundaries in this solution. So two things to note from this version of the model without activity. One is that we had a long time solid viscoelastic material, but now solved on a moving domain. And it shows this yielding kind of behavior because the constitutive laws have now become implicit in the movement of the domain. Second is that even without activity, this model shows something that reminds us of determinate, indeterminate growth, depending on the ratio of this applied boundary stress and the elasticity of the material. So what happens with activity? Again, the two phases persist. You have something that grows for a while and stops, and something that keeps growing. But now the role of activity is, the role of the applied boundary stress is played by the active stress at the boundaries. Again, one can compute the growth rates in the perennially growing phase and the final length in the stopping phase. The growth rate acquires some correction due to the gradients in the density field, which clearly they will be minimized when kappa is large, which is what we see. So these are growth contours. The red curves are the contours of this growth rate, and the green curves are the contours of this final length. And at large kappa, the numerical contours of the growth rate match the one given by this boxed equation. In 2D as well, the two phases persist, but there is no shape change unless you prescribe some heterogeneity in the initial condition or material parameters. And finally, the last thing about this model is that at low kappa, this starts showing tissue scale oscillations. And that essentially arises when you choose an active stress that has both a contractile and an extensile part, because you have both birth and death in the density equation. Yeah, so, and if you include the orientational degree of freedom that you neglected, that comes from basically oriented divisions or orientation in cell intercalation directions, you see many non-trivial phases coming up where this is depending on the correlation length of the pneumatic and the hydrodynamic length of the tissue, you start seeing very, very non-trivial behavior. So I'll stop here and be happy to take questions.
You have time for one very quick question. I'll ask the question. So, do you see any Fisher wave type behavior?
No, we don't see. That's only time-dependent solution, only oscillation.
Let's thank the speaker again.